morning everyone. First of all, I should congratulate the team of Rishikesh, Girish Babu and Co for organizing such a fantastic conference, which was our dream in our PG days to have such a conference. So today I am speaking about the important neurotransmitters. Before starting going to the topic, I start from the basics, because most of the time we don't know the basics, then we try to understand something we'll never understand. So neurotransmitters, neurotransmission happens in the neuron. Neuron structure is like this: there is a cell body, there is a dendrite, there is an axon, and the neurons connect to each other to form synapse. You can see here an axon of many different neurons all arriving to form a synaptic connections with the dendritic tree of postsynaptic neuron. Okay, the question is, what constitutes a neurotransmitter? Many times you would have seen in the exams, what is a neurotransmitter? First paper 1 question is, what is neurotransmitter? What does it contain? Apart from the exam, even for the understanding of any of the neurobiology, we should be sure about the definition of neurotransmitter. Basic criteria to call something as a neurotransmitter is that it is synthesized in the neuron and it is released from the neurons. Secondly, it is released in the nerve terminals in a chemically and pharmacologically identifiable form. Third thing, it interacts with the postsynaptic receptors and brings about the same effects which are seen in the stimulation of a presynaptic neuron. It interacts with the postsynaptic receptor, displaying a specific pharmacological profile, and its actions are terminated by the active processes. Finally, a neurotransmitter is the one which is synthesized and released by the neuron in a pharmacologically identifiable form, which acts on the postsynaptic neurons to produce some pharmacological actions, and its actions are terminated by active processes. Okay, how do we classify neurotransmitters? Neurotransmitters are classified based on their chemical structures like amines, pituitary peptides, circulating hormones, amino acids, oral hormones or opioid peptides, genes, lipid neurotransmitters, and also gaseous neurotransmitters. But I am not going into all those things. My major focus today is on the biogenic amines like dopamine and serotonin, amino acid, specifically glutamate. These are the 3 neurotransmitters which I will be concentrating today. Neurotransmitter is released from the neuron. It should act with the receptor, right? What is a receptor then? The basic essence of all living cells is that it should recognize the external event and respond to it adequately. Similarly, the 2 major functions of the receptor is that recognition of specific molecules and activation of effectors. Okay, the neurotransmitters can be. Where are these neurotransmitters synthesized? We always talk about the receptors getting down-regulated, up-regulated, but basically, where these neurotransmitters come from? As monoamines are synthesized in the cell bodies, even the neurotransmitters are synthesized in the cell bodies, then they are transported in the axons to the nerve terminals. The neurotransmitter, based on their location, can be simply classified into autoreceptors and heteroreceptors. The definition runs like this: receptors which are located on the neuron that produce endogenous ligand for that particular receptor. I'll make it simple. A serotonin receptor on the serotonin neuron is called an autoreceptor. If a serotonin receptor is present on a dopamine neuron, we call it a heteroreceptor. And based on the mechanism of action, the neurotransmitter receptors can be classified as inotropic, G-protein coupled, receptor tyrosine kinases, and nuclear receptors. To make it very simple, inotropic receptors are those receptors, they act by the ion channel closing or opening. Well, the G protein coupled receptors act through the second messenger system. The tyrosine kinase act through the phospholipase C activation and phosphorylation or dephosphorylation. What is this receptor regulation? We always talk about depression. We start an antidepressant. He doesn't respond on the day one, but he responds after 10 or 14 days because the receptor down regulation should happen, right? We always write and read these things. What is this receptor regulation? What do we mean by that? When there is neurotransmitter depletion, the number of receptors are varying. Sometimes there may be increase in the receptor or decrease in the receptors. Once we start a drug, maybe a SSRI or TCA, there are some intracellular signal changes which directs this DNA to either increase the receptors or decrease the receptors. To make it very simple, in the above you can see there is down regulation, means the number of receptors are decreasing. In the down figure you can see there is up regulation of receptor where there is more receptors are synthesized. Coming to the real today's topic, the serotonergic system. Why did serotonin get its name? Serotonin was identified to be an endogenous vasoconstrictor in the serum. That's why the name serotonin. Serotonin. It has an inbuilt endogenous character of vasoconstriction. And where it was later acknowledged that what we had studied in the physiology, the secreted in the intestinal cells is nothing but serotonin. So, where this serotonin gets synthesized? It gets synthesized in the raphe nucleus. What do we mean by raphe? Raphe in the Latin means middle. You can see the raphe nucleus in the picture. This raphe nucleus secretes serotonin. How this serotonin gets secreted in the cell bodies of the raphe nucleus? Basically, the precursor for serotonin is the tryptophan. The tryptophan enters the neurons. Tryptophan gets converted into 5-hydroxytryptamine by the enzyme tryptophan hydroxylase. After that, 5-hydroxytryptamine gets tryptophan gets converted into 5-hydroxytryptamine. Tryptophan gets converted into 5-hydroxytryptophan by aromatic amino acid decarboxylase. What happens after that? It gets collected in the vesicles, then gets secreted in the synapse. After that, they get attached to the transporters going back into the neuron and get metabolized by the monoamino oxidases. Then coming to the serotonin receptors. You know, there are many number of serotonin receptors, up to 30 to 14 serotonin receptors. Like starting from 5-HT1, 2, 3 to 7. Among them, there are a lot of subtypes. Before going into the types of serotonin receptors, I want to discuss about the various pathways in the serotonin system. You know, I was talking about that it gets secreted in the raphe nucleus. From the raphe nucleus, the connection to the cortex. This circuit majorly plays a role in the regulation of the mood. The connection to the basal ganglia from the raphe nucleus is implicated in terms of OCD, agitation, akathisia. If you read the Maudsley guidelines, in that flow chart, you can see mirtazapine is one of the choice for akathisia. You know that akathisia is induced by antipsychotic. Why is this? This is because 5-HT1A receptors are in the presynaptic neurons. These, if it is get stimulated or agonistic action on the 5-HT1A receptors, decreases the serotonin transmission. So, mirtazapine, it is a specific adrenergic. It is a non-adrenergic specific serotonergic antidepressant. It has a different kind of action where it is a stimulant of 5-HT1A receptor. The Connections from the raphe nucleus to the limbic system is implicated in anxiety and panic. And to the hypothalamus, it is implicated in appetite and bulimia. The projection from the raphe nucleus to the chemotrigger zone is implicated in vomiting and nausea. The spinal cord, the projections from raphe nucleus to the spinal cord is implicated in the sexual functioning. You know that most of the serotonin is against sexual functioning. You know one of the most common side effects of SSRI or TCA, sexual dysfunction. But a newer antidepressant came called as vilazodone. Most of you have heard about that. What is vilazodone? Vilazodone is a sparring, that is serotonin, partial agonist of serotonin. Where what partial agonist is about the 5-HT1A. If it is partially agonizing the 5-HT1A, so the serotonin transmission becomes less, so that the sexual dysfunction is less, and at times people who had sexual dysfunction from other antidepressants can be corrected using these antidepressants. Let us speak about the individual receptors. The 5-HT1A receptor, which is in high density in the limbic system. The speciality about this receptor is that, like we know, like when we give that antidepressant, or whenever the raphe nucleus is damaged, or when we give some of the serotonin antagonists, serotonin depleters like PCP, usually the receptors get either down-regulated or up-regulated. But this receptor is not sensitive to any of these. This receptor is more sensitive to the adrenal steroids. Any adrenal steroid will have a definite effect on this 5-HT1A receptor. You can see in the picture, as I told you, if the receptor is in the cell body and the dendrite, the somatodendritic receptor, whenever this 5-HT1A presynaptic somatodendritic receptor recognizes serotonin, you can see in the picture, in the second picture, it will actually shut down the neuronal transmission. 5-HT1A is the autoreceptor which is located in the somatodendritic serotonin neurons. If it recognizes a serotonin, it will shut down the neuronal transmission. Then coming on to 5-HT1D and 1B receptors. 5-HT1D receptor is majorly in the axon terminal, and these 5-HT1D and 1D receptors inhibit cyclic AMP and it forms and stimulate IP3 production and phosphokinase C. Again, you can look at this. It is in the axon terminal 5-HT1D receptor. If serotonin occupies this 5-HT1D receptor, there is blockage of serotonin transmission. Then about which are the heteroreceptors which are significant, which are located in the serotonin neurons. Know about this alpha-2 receptors. Whenever this alpha-2 receptors present in the presynaptic serotonin neuron, if the neighboring noradrenergic neuron produces this norepinephrine and sit on this alpha-2 receptor, it is inhibitory for serotonin transmission or release. But in contrast, the alpha-1 somatodendritic receptor, which is on the serotonin, accelerates the serotonin release. Then coming on to the 5-HT2A receptors. Most of the time, we see the antidepressant going and sitting on the 5-HT2 receptors. Lot of hypothesis is coming telling that the major implication of 5-HT2A receptors in the causation of depression. If you administer for a long time antidepressants, it will down-regulate the 5-HT2 receptors. But in contrast, when you give electroconvulsive therapy to a patient with depression, there will be upregulation. There will be more 5-HT2A receptors synthesized. So you can see once the 5-HT2A receptor, once the serotonin sits on the 5-HT2A receptor, it will increase the neuronal impulses in the serotonin neurons. Coming on to the 5-HT3 receptors, you would have heard about ondansetron. Ondansetron is an antiemetic used in chemotherapeutic patients. The basic mechanism of action of ondansetron is blockage of 5-HT3 receptors. It is a ligand-gated receptor. It mediates fast neuronal responses. Why it is used as an antiemetic is that its location is in the chemotrigger zone, which includes the area post-tremor. The other point about ondansetron is that, about 5-HT3 receptors is that it gets modulated by different drugs, alcohol and anesthetic agents. So even you could have read that ondansetron can also be used as an anti-craving drug for alcohol. The 5-HT4 receptors are actually apart from 5-HT1A, 1D, 5-HT2, 5-HT3, later 5-HT4, 5, 6 and 7, not much of literature available about these receptors, but there are some points to mention about 5-HT4 receptor. Basically, it modulates the release of monoamines and gamma-aminobutyric acid in the brain. The 5-HT5 receptors are located in hypothalamus, hippocampus, corpus callosum, cerebral ventricles and glia. Look at this 5-HT6 receptors. It is situated in the nucleus accumbens. All of you know nucleus accumbens is a reward center. So any anti-craving drug to be developed targets the nucleus accumbens. So 5-HT6 receptor may be targeted later. That 5-HT7 receptors, here it is located in the cerebral cortex, thalamus, substantia nigra and dorsal rofin nucleus. They have seen chronic treatment with antidepressants. They have shown that 5-HT7 receptor gets down-regulated, but acute stress increases the expression of 5-HT7 receptors. The neurotransmitter receptor hypothesis of depression. What is this? Like we tell, there is imbalance in the monoamine, so there is depression. This is the monoamine theory about depression. But when you give an antidepressant, the day one itself, there is increase in the serotonin or norepinephrine, whichever the drug you give, there is increase in the synapse of these neurotransmitters. But the patient doesn't respond. We always educate the patient that yes, we have started today, it may take 1 or 2 weeks to you to feel the improvement. So how the hypothesis of monoamines can be accepted? So the next hypothesis came, it is the monoamine receptor hypothesis. This hypothesis speaks about when depression is there, it's more of the receptor imbalance rather than the neurotransmitter imbalance. In the beginning of the talk itself, I showed you the receptors, what is down regulation and up regulation. That I wanted to make the point, what is this down regulation? It's basically the down regulation or the regulation of the receptors which is implicated in the causation of depression. Next I am going to the glutaminergic neurotransmission. In the 1970s, both the dopamine hypothesis and also the NMDA hypothesis was in the parallel lines for the hypothesis of psychotic illness. I don't know for the reasons, but the glutaminergic hypothesis never took the lead. As we all know, glutamate and aspartate are the two major excitatory amino acids in the CNS. Glutamate appears to play a very significant role in synaptic plasticity, learning and memory. How is glutamate produced? It is basically produced from glucose and also transamination of ketoglutarate. The small proportion of glutamate is directly formed from glutamate. You can see this picture, the glutamine from the glial cells enter to the neuron. There the glutaminase enzymes coming to the picture produces glutamate. glutamate is removed, but not by metabolism. It is more of the transporter system which is involved in the removal of glutamate. Glutamate has three receptors. One is the NMDA receptor, ampakines and kynurenate receptors. So, you know about this neurodevelopmental as well as the neurodegenerative model of schizophrenia. According to this theory, to brief, it is a neurodevelopmental disease, and also the neuronal degeneration progress very fast. Even since the beginning, it was called as dementia precox, because the dementia, that's the neuronal loss, sets in an early age. There comes the role of this glutamate receptors. This glutamate receptor, we'll talk about the NMDA receptors. NMDA receptors, like you know that GABA benzodiazepine receptor. Here, glutamate calcium receptor is there. You can see this in the picture. There is a site for glycine, there is a site for polyamide, and also there is a site for PCP. PCP name is given to you. This drug acts on this point. So, what happens in glutamate? Once that it is an ion-gated channel, once the opening of the calcium channels is there, once the calcium ions enter, there will be excitation. The excitation to certain extent is good. If the excitation goes run amok, it leads to excitotoxicity. There is excess of calcium flow into the glutamate receptors, leading to the formation of enzymes. These enzymes in turn produces free radicals, which causes the destruction of each of the cells in each of the components of the cells, neuronal cell. So here, why this is important? One point is that we know that there is positive symptom, there is negative symptom. Positive symptom is because of lesser dopamine or lesser excitation. Negative, more excitation. Negative symptom is lesser dopamine, lesser excitation. So, why can't we find a drug which can cause stimulation through the glutamate? We spoke about the one phase of glutamate where it gets excited, it causes toxicity. Other phase is that there is a drug which has been developed called as pitocteri. What is this pitocteri? Now we want the negative symptoms to go, means we want something which keeps the person active. For active, you should need some sort of excitation, but not to the level of excitotoxicity. So, if at all, if you give glutamate, you know that it will cause us excitotoxicity, neuronal damage and all. We want the NMDA receptor to be activated to a certain extent so the negative symptoms will disappear. I showed you a picture here. It also has a site for glycine. The newer drug which was developed and has got English FDA approval works on this glycine. We know SSR, selective serotonin reuptake inhibitor. Similarly, this glycine reuptake inhibitor, so that the glycine is preserved in the synapse and it acts on the NMDA receptors, causes activation but not toxicity. You are all bright young individuals, lot of hope in you, so that you can develop some drug where you know every time when you give an antipsychotic, delusions, hallucination disappear. But what about the negative symptoms? There comes this pitocteri. So other theories about like I told you, NMDA, if it gets stimulated, goes into excitotoxicity. So the drugs for NMDA antagonism also helps in controlling. Then I come to the dopamine. All of us always start our psychiatric career with studies on dopamine. Now, dopamine is produced in the neuron. The precursor for dopamine is tyrosine. Tyrosine hydroxylase converts dopamine to Dopa carboxylase converts dopamine into dopamine. As I told you, it gets into the vesicles, secretes in the synapse, and after that either it will be taken back through a transporter or metabolized by catechol-methyl transferase or monoamine oxidase. So how many monoamine dopamine receptors we need? D1, D2, D3, D4, D5. Since D1 and D5 matches in certain structural perspectives, so we call it as D1 family. The second is the D2 family, D3 and D4. Why D1 and D5 are in the same group is that both stimulate adenyl cyclase activity via activation of G proteins. The D1 is mostly located in the frontal cortex. So we know frontal cortex more of cognitive function. So D1 receptors are more important role in cognitive functions. D5 receptors is a neuron specific receptor that is located primarily in the limbic area. D2 receptor. This is the one which opened the eyes of the psychiatrist once the conventional antipsychotics were launched. They block the D2 receptors. We have two types of D2 receptors where one the shorter and the long form. The shorter form is D2S and the longer form is called the D2N. It works through the inhibition of adenyl cyclase. D2 receptors are actually located in the cell bodies in the nerve terminals of dopamine neurons. Activation of the somatodendritic D2 receptors reduces dopamine neuronal firing. The somatodendritic near the cell body, D2 receptors are there. The dopamine sits on that. It decreases the firing of the neuron. Activation of the in other ways, those dopamine D2 receptors present in the axon terminal reduces the amount of dopamine released from the neuron. So the D2 receptor in the somatodendritic area reduces the neuronal firing, while the D2 receptors in the nerve terminal decreases the dopamine released per nerve impulse. The D3 receptors are preferentially in the limbic area. It is maybe a target for the future antipsychotics. One more point about D3 receptor is that the BDNF, the brain-derived neurotrophic factor, regulate behavior sensitization via D3 receptors. D4 receptors, clozapine has a very high affinity for D4 receptors. D4 antagonists are used as adjunctive agents in treatment of schizophrenia. Genetic D4 variants may be talked about novelty seeking, ADHD, all these things. They have found some genetic variants in D4 receptors. You know the four well-known the dopamine pathway: there is mesolimbic, mesocortical, tuberoinfundibular, and nigrostriatal pathway. Mesolimbic pathway from the ventral tegmental area to the limbic system, mesocortical from the ventral tegmental area to the cortex, the nigrostriatal from the substantia nigra, and tuberoinfundibular from the anterior pituitary hypothalamus. So we know that D2 receptors are blocked by the conventional antipsychotics. Apart from blocking the D2 receptors, our conventional antipsychotics also block the muscarinic M1 receptors, which led to the unnecessary side effects of constipation, blurred vision, dry mouth, and drowsiness. I want to focus more on this slide. Why does extrapyramidal side effects happen? What is the connection between acetylcholine and dopamine? Dopamine actually stimulates acetylcholine. Once dopamine is blocked, that will cause us anticholinergic. Apart from decrease in the dopamine, it is the severe anticholinergic effect which produces EPS. That's why the first choice of drug is tricyclic. Our conventional antipsychotics also blocks the H1 receptors. It causes weight gain and drowsiness. Alpha-1 receptors are also blocked, which causes variation in blood pressure, dizziness, and drowsiness. This slide, like most of the off late, we are not seeing this tardive dyskinesia. Couple of weeks back, I saw one of my neighbor having severe tardive dyskinesia. We were never aware. She was on flupentixol for 25 long years. Why this tardive dyskinesia happens? That's where again this down regulation or up regulation comes into picture. When we are giving antipsychotics for a long time, this causes continuous blockade of this D2 receptors. So that upregulation of this D2 receptors will happen. You can see in the picture. That causes dopamine supersensitivity, leading to the tardive dyskinesia. Then we'll talk about second generation antipsychotics, also known as serotonin dopamine antagonists. We know that second generation antipsychotics block lesser proportion of dopamine, so that also contributes to the lesser chances of extrapyramidal symptoms. More than that, they block 5-HT2A receptors. What is the connection between the 5-HT2A receptors and the dopamine? You can see this the substantia nigra circuit. Here the raphe nucleus is there, substantia nigra is there. They are projecting into the cortex. Like the serotonin has a brake-like effect on the dopamine neuron. Means you are driving a car, you are applying brake. You are applying brake, but you are not ready to release the brake, but you want the car to move. What we can do, if we paralyze your leg, suddenly the car will move. Similarly, the serotonin receptors, 5-HT2A receptors, acts like a brake on the dopamine receptor, dopamine neurons. Once you block that 5-HT2A receptor, dopamine get released better and lesser chances of extrapyramidal side effects. You can see in this picture, not only here, again the dual action comes because you are blocking the 5-HT2A receptors, so there is more dopamine. Apart from that, already some dopamine receptors are blocked and there are more dopamine, there will be more computation for the receptors. So the extrapyramidal symptoms decreases. This I am talking about the nigrostriatal pathway. You may ask, if at all this reversal happens in mesolimbic pathway, what is the use? Fortunately, this doesn't happen in the mesolimbic pathway. Otherwise, the basic function of any of the antipsychotic is to block the 5-HT2 receptor in the mesolimbic pathway, so that there will be the psychotic symptoms will decrease and the patient will improve. But we are targeting the extrapyramidal symptoms, so it happens only in the nigrostriatal pathway as well as in the mesocortical pathway. We know that mesocortical pathway hypodopaminergia causes all these negative symptoms like apathy, evolution, and all these things. So here, in the mesocortical pathway also, atypical antipsychotics of the second generation of the serotonin dopamine antagonist increases the amount of dopamine and decreases the negative symptoms also. This is one interesting emerging model. Like I was all along talking about why negative symptoms, why positive symptoms, what is the connection between the glutamate, the dopamine, GABA and all. There is somehow this emerging model says that there is some dysfunction in the NMDA receptor or the hypofunction in the NMDA receptor, where the dopamine release will be more in or more than dopamine, the dopamine release will be more in the mesolimbic pathway. But similarly, in the mesocortical pathway, there will be excitotoxicity and lack of neurons and destruction of the neurons. So, what all concepts we have learned all these while are a bit shaky. The another newer concept is that it's like a person goes with a cough to a pulmonologist. a similar person come to us with the symptom of hearing voices. We might have brand him psychosis, start an antipsychotic. But a person who goes to a pulmonary, get an X-ray done, it may be because of tuberculosis, it may be because of pneumonia, it may be because of even the cancer, small cell carcinoma. That X-ray can differentiate. Similarly, I'm optimistic in another 10-15 years, we have some sort of the imaging will be done. It's all because of the loss of the grey matter. Loss of the grey matter may be because of the excitotoxicity or the neurodevelopmental things. So, somewhere in the recent future, we may get a neuroimaging techniques so that, Masheri Keshwan in the recent PCMA in the January issue, you could have seen about research advances. The whole findings of the B-SNP study have been published. So, we can develop an instrument so that we can some tools so that we can classify them as psychotic or neurotic, or what is the treatment required and optimize them for that. As I told, the newer targets about the glutamides, like the metabotropic receptors have been one drug was developed, but it did not show much improvement. As I told, the glycine which was used, I was also involved in that trial, and the trial showed the trial included not full positive symptoms less and more of negative symptoms. We could see a good, in the initial trial, placebo effect was more, so the trial was dropped. Again, it has got US FDA approval to be used in negative symptoms of schizophrenia. Thank you.